Hello guys, welcome sa final topic natin on our midterms. We have recombinant DNA technology. Get your drink, your snacks. So this is a long ride pero marami tayong matututunan. So we'll talk about the different terminologies in regards to recombinant DNA technology. What are the steps? Merong 5. And within that steps, guys, we will discuss the different tools para ma-accomplish yung mga steps like yung scissors natin, restriction enzymes, yung glue natin, DNA ligase, our vectors. So these are your plasmids, where your desired gene will be inserted, modes on how to transfer the gene and to detect the gene. Yan. So, mga 40 slides to, kaya natin to. I'll divide the PowerPoint into 2. So guys, ang isa sa pinakamagandang resulta, talagang groundbreaking result of recombinant DNA technology was the production of factor 8. So factor 8 is missing among patients who have hemophilia. So madaling mamatay dati ang mga patients with hemophilia. They could die because of internal bleeding because of the lack of factor 8. At nung around the 1980s, nag-racing itong Genentech Company and Genetics Institute. So they were racing to find the how to produce factor 8 artificially through recombinant DNA technology. Yan. So meron akong picture from Genetics Institute, from GI, si J Tool. Yan. So Genentech, I forgot date. Wala akong nakitang photo nila eh. So nagko-compete itong dalawang company to. So what were the problems that they have to overcome to create factor 8 recombinant drug? So back then, 1980s, there's not enough knowledge on factor 8. Hindi ba, just a review, your factor 8 is attached to von Willebrand factor. So if you will only purify or produce factor 8 on its own, it's scarce in the body and it is unstable without the von Willebrand factor. And back then, guys, the only treatment for hemophiliacs for factor 8 was blood transfusion. At maraming hemophiliacs ang biglang nagkakaroon ng HIV. Kasi guys, hindi pa uso ang blood transfusion screening noon. So, tumataas rin ang hemophiliacs na may HIV. Grabe na yung mga problems that you have to overcome. And guys, compared to insulin and growth hormone, ang insulin, maliit lang pala siyang na molecule. It only has 51 amino acids and growth hormone has 191 amino acids. They created already humulin, yung recombinant form ng insulin. So, na-produce na nila artificially. Pero, so, what they have to do now sa factor 8, ang haba ng base pairs ng gene ng factor 8, 186 kilobase pairs. It's a very big, sabi nga sa binasa kong history, it's a gargantuan molecule with 2,351 amino acids. It's a big molecule that they're trying to create. Yan. So, ganda ng story nila. And guys, in I don't know what the year is, but guys, they had a photo finish. Ibig sabihin, sabay, almost the same, na nagkaroon ng patent ang Genentech and Genetics Institute with factor 8 recombinant drug. Okay, so halos sabay sila nagkaroon ng patent. And I think hanggang ngayon is they're fighting kung sino nauna sa patent. Yan, so ang galing, no? So that's your anti-hemophilic drug. So bawat recombinant drug, guys, is talagang pinag-iisipan. It took a very long time. For 2 years daw sila, they were working non-stop, 16 hours a day, 20 hours a day, kasi ang taas ng, they were raising, taas ng investment dito in making the recombinant drugs. Galing, no? So first terminology that we're going to discuss is your genetic engineering. So genetic engineering is the umbrella, yan, under where your recombinant DNA technology is. Yan. So, process of using, so yung genetic engineering, tool niya, itong rDNA, to alter the genetic makeup of an organism. So, kumbaga, if we want to be technical, pero sometimes I'm using them na pare-pareho yung meaning nila. Pero genetic engineering, yeah, we engineer genes, and one tool that we do is recombinant DNA technology. So this is a technique. Yan. So rDNA, ang other name niya, to identify, isolate, and insert the genes of interest into a vector. At talagang pag-uusapan natin yung halos an overview lang to of how is rDNA possible. Yeah. So our goal is to produce large quantities of the protein that the gene produces. So example, so yung insulin gene, yan, our goal is to create a lot of artificial insulin from recombinant DNA technology. So yung kakadiscuss ko, factor 8. So nakaproduce sila ng mass production ng factor 8 para sa mga hemophiliacs natin. And gene cloning or molecular cloning. So itong molecular cloning, guys, it's under your recombinant DNA technology. So ito, kung daw nagkakamali, ito yung 5th na step, paparamihin mo yung genes. Yan, para you could harvest the protein that it produces or that it expresses. So guys, ito ay ang ating mga transgenic organisms or genetically modified organisms. So, wala silang masyadong, they're genetically modified because nag-add ng green fluorescent gene sa kanila para mag-produce sila ng gene fluorescent protein. Yan, that's it. So, ito yung bunnies, fruit fly to, mice, zebrafish. I think this is a seaweed. Yan. So, again, they are inserting genes in these animals. So, yun. Para magaling the dark. Walang masyadong use yan. Later, I give example yung may use. So, as an overview of the different steps, basically talagang pag-aaralan natin how is this possible. We will add more details, yung buong process niya. So, this is your plasmid, also called as your vector. Yan. And we have this foreign DNA. Sabihin natin itong DNA na to, this is where your gene of interest is. May gusto kang gene na gusto mong paramihin. Yan. So, basically, you cut it using your restriction enzymes and insert it into your vector or your plasmid. Yan. So you could now multiply your plasmid. Pwede mo na siyang paramihin. Or you first, mali pala, you have to insert it pala. Introduce it to a host. Example, talagang pinaka-common, bacteria. So we will also discover that plants, pwede rin tayo mag-introduce ng bagong gene sa kanila. So, yan, pwede nating paramihin yung bacteria. So, mapaparami na rin yung desired gene natin. So, mapapansin nyo dito sa image, ito yung desired gene natin, yung plasmid with the desired gene. So, ito yung mga bacterial cells na na-transform or naging competent. Or tapos, ito yung mga bacterial cell na hindi naka-uptake or they were not transformed. Yeah. Tapos paparimihan mo sila. So we will have tools to detect which one had the gene or not. Yeah. So actually, na-discuss na natin itong mga to na pakonti-konti. So today, pag-aaralan natin yung buong picture. Okay. Sorry guys, hindi muna ako. So steps on rDNA technology. So step 1, so we need to identify and isolate the gene of interest or the DNA fragment to be cloned. So ano ba yung mga sources natin? So then we will insert the gene to a vector. So pag sinabi na vector, mga plasmids, bacteriophages, later we will talk about it. Then, introduction of vector into an organism or cell or a bacteria to transform it. Then, selection of the transformed cell. So, alin doon yung nag-express ng gene and we need to multiply it. So kung na-insert mo na yung vector, yung plasmid sa isang bacterial cell, we need to multiply it. So simulan natin sa step 1. So identification and isolation of gene of interest. Yan. So where are the sources of our genes of interest, ma'am? Yan. So saan ba natin kinukuha yung mga ini-insert natin sa vectors natin or plasmid? Actually, we have a DNA library. Wow, di ba? So if the library is a place where you could get a collection of different books, meron din tayong DNA library. Di ba? Nag-establish na sila ng ganun, wherein we could get our different collections of DNA fragments. Yan. Lahat ng mga DNA na kailangan mo nasa DNA library. You could now just order it. Chemical synthesis. If you know the amino acid sequence, you could synthesize pala yung genes. And guys, if you have a portion already of the gene, you could now amplify it. Kung gusto mong paramihin pa or pahabain pa, you could now use a PCR machine. Okay? For this topic, we'll focus on DNA library. Okay? So there are two types of DNA library. We have genomic DNA library. Pag sinabi natin genome, lahat na talaga. So kasama na doon all the genes in the human genome. Yan. So how it consists of introns and exons. So it consists of both the coding and the non-coding regions. So ibig sabihin, yung mga genes na nagko-code, yung may mga silbi, sabihin natin, at pati yung mga blankong genes, yung mga walang use, kasama rin. So both introns and exons. So dito sa cDNA, complementary DNA library, exons na lang. Yung mga coding regions na lang, yung mga mahalagang regions na lang in contrast to that. So ma'am, bakit ang pangalan nito complementary DNA library? Actually, these were mRNAs. Yan, mRNA siya. That they added the reverse transcriptase enzyme para maging DNA form. Yan. So these are mRNAs that were nag-add ka ng reverse transcriptase para maging complementary DNA. So they were formed from the addition of the reverse transcriptase. So guys, ano bang advantage nito? Yan. Ang advantage niya, it only consists of exons. If you remember guys, in the process of transcription, diba? Bago lumabas yung mRNA, it will be further processed. And we have this term spliceosome, meaning the removal of the introns, yung mga non-coding regions. Yan. So pag mRNA, mas pulido, mas short, yan, it contains few base pairs only. Yan. So maraming talagang reasons kung ba't pipiliin ito scientists, kung pipili sa genomic library or sa cDNA library na lang siya. Kasi pag sa genomic library, talagang yung buong gene ang makukuha niya, even the introns. Kung may gamit siya sa introns, di genomic DNA library ang i-search niya. However, kung gusto niya lang talaga yung mga coding regions, they could get their DNA from your cDNA library. Galing no, guys? So, ang pagkakaiba nila, puro exons lang to, ito, introns and exons. If you want to get the complete genes, go to genomic DNA library. If you just want the coding regions, cDNA library. Okay? So, these are actually mRNA na nalagyan ng reverse transcriptase. So, kayo, yan, kung gusto mo ng genomic DNA or mRNA, so parang mRNA, para maging cDNA, add reverse transcriptase. Yeah. So you could now, at saka guys, ang advantage kasi, ang mRNA, kinonvert siya sa cDNA because kung gusto mo siyang paramihin, okay lang. Kasi ang PCR guys, ang kaya niya lang i-amplify are DNAs. Okay? It cannot amplify RNAs. So if you want to amplify RNAs, you have to convert it first to cDNA. Okay? So in our SARS-CoV-2 PCR testing, kaya ang pangalan niya RT-PCR because your SARS-CoV-2 are RNA viruses. So, you have to add reverse transcriptase para maging complementary DNA yung virus mo para ma-amplify mo siya. Okay? So, hindi na-amplify ang RNA. Okay? You have to convert it to complementary DNA. So, kung yung scientist gusto niya i-amplify, pwede-pwede. Okay. So, insertion of isolated gene in a suitable vector. So, first, we have to isolate the gene. So, syempre, para ma-isolate mo yung gene, kailangan mong guntingin, di ba? Kasi gusto mo lang yung gene of interest mo. So, we will discuss now our restriction enzymes. After that, yung glue naman, para mapunta yung gene of interest mo dun sa vector, you have to glue it with DNA ligase, and we'll talk about the vectors, kung yung the suitable vectors. So, plasmid, bacteriophages, cosmids, bacterial artificial chromosomes, and yeast artificial chromosomes. So, yan ang mga next topic natin. Kasi nasa step 2 na tayo. So, isisingit ko na tong tatlo. Yeah. Okay. So, let's talk about your restriction enzymes. Ma'am, saan mo na-invento to? Meron ba sa tao nito? Do humans have restriction enzymes? None. Okay? The only sources are your bacteria. How did they discover this? Ang galing-galing. So, for a million years, guys, magkaaway ang bacteria at bacteriophages. They're fighting each other. Ang goal ng bacteriophages is to hijack the bacteria. So, gusto nilang i-hijack yan because they want the bacteria to be a host para rumami yung bacteriophages. Para makapasok siya sa loob, hijack it, tapos magmumultiply yung bacteriophages sa loob hanggang pumutok yung bacteria, tapos rarami, magmumultiply yung bacteriophages. So, bacteriophages are viruses, okay, that attacks your bacteria. Okay, so they're for millions of years naglalaban sila. So what do the bacteriophages do to hijack your cell, bacterial cell? They inject their DNA dito sa bacterial, sa chromosomal DNA natin. Yan. So, mapapansin nyo, may mga red na portions dito. So, yan yung mga in-inject na foreign DNA from your bacteriophages. Yan. So, what the bacteria does to protect itself, yan, para ito protect yung mga essential na chromosomes, is it will methylate it. Yan. Andito yan. Wala ata. It will methylate their DNA. Yung mga mahalaga, yung sa kanya, yung mga chromosomal DNA niya, i-methylate niya. What does methylating mean? You will add methyl groups. The E. coli will add methyl groups to the existing bacterial chromosomal DNA. Okay? So, hindi methylate yung mga foreign DNA, yung mga red. Okay? And this bacterial enzyme will now produce a restriction enzyme or a scissor, okay? And this bacteria will cut off all the foreign DNA. So, yung mga methylated portions lang ng chromosome, yun yung maproprotektahan. Yung mga non-methylated, which are your foreign or viral bacteriophage DNA or viral DNA, yun ang magugupit ng restriction enzyme. So, doon nila na-discover itong restriction enzymes, guys. How the bacteria protect itself. So, by methylating its bacterial chromosome and by producing a restriction enzyme that will cut all the foreign DNA. Ang galing, no? And so, na-discover ang restriction enzyme. Can you search? Sino naka-discover ng restriction enzyme? Nakalimutan ko ilagay. So your restriction enzyme, ang specific na pag-aaralan natin are restriction endonucleases. So this restriction enzyme cuts within the nucleotide sequence, sa gitna. Pag sinabing restriction exonucleases, sa dulo, end of the nucleotide region. So, mag-focus tayo sa restriction endonucleases. So, these are proteins that cleave DNA molecules at specific site. Yan. So, each of your restriction enzymes will detect a certain DNA sequence. Okay, example, ito, BAMH1, yan. So, or BAMHI ba to? Wait. Alam ko, ay yan. So, pag na-detect na itong GG80CC, yan, and its complementary base pair CCTAGG, it would cut it in such a way na mukhang step ng stairs. Yan. So, specific yan na. Ito lang talaga yung paggupit niya. Yan. So, this is the end fragment and it will produce a sticky end. Okay? So, your restriction enzymes could either produce a blunt end, yung derecho, or a sticky end. So, other notes, sabi, cohesive end or protruding ends. Yan. So, this is a sticky end or protruding end if there is a overhang. May extension or also called as a overhang. Yan. So, they say that, it has a better, this type of cutting is more stable, okay, compared to blunt ends. Yan. So, pag blunt ends, diretso lang. So, sticky ends, is mukhang, step ng stairs. So, for this, eco R1. So, GAATTC naman yung sequence niya na hinahanap niya. So, itong sequence nila, guys, ito lang ang ikakat nila. Okay? So, tanging ito lang ang ikakat nila. So, I'll give a little bit of restriction enzymes. Yan. So, at some point, kailangan yung aralin, okay, at i-memorize kung ano yung mga kinakat nila. So, itong BAMHI, GATCC, ito, GAATTC. So, ito yung sequence na kinakat ng eco R1. And notice, sticky ends rin yung pinoproduce niya. Okay. So, let's give more examples. Itong HIN3. So, HIN3 yan kasi may HIN2, may HIN1. So, sticky ends rin. Sa PST1, sticky ends rin. Sa eco R5, we have blunt ends. Yan. So, guys, notice where the overhang is. So, notice, guys, na itong HIN3, ang overhang niya is 5'. Yan, 5'. So, 3 yung nandito, 5. Yan, sa 5'. So, yung overhang ng PST1 sa 3'. Yan. Tapos, blunt ends naman dito. So, guys, pag magkakat ka, kunyari, magdidikit ka na ng gene of interest and your plasmid or your vector, kung gusto mo magdikit sila, dapat you should use the same restriction enzyme. So, example, in your foreign DNA. So, cut mo siya using eco R1. Yan. Cut mo siya yung foreign DNA. Yung vector mo rin, i-cut mo rin gamit ang eco R1 so that pag they would be complementary to one another. Okay? So, AT, GC, CG, T, ano naman complementary ba siya? Ayan, TA. Yan, tama naman. So, itong pag blunt ends, hindi na mo kailangang complementary base pair. Diba? Notice? Hindi na kailangan. So, no need to use the same restriction enzymes. Pero pag protruding ends or sticky ends, you have to use the same restriction enzymes. For more example, we have ALU1. So, itong mga names, guys, it's from the bacterial sources. Yan. So, notice like yung mga blunt ends. Itong mga blunt ends, AL1, A3, yan, BAMHI, BAM1, BAMH1, I think. Ayan, so mga blunt naman yan. So, notice, pagkaiba talaga yung mga protruding. Ito, 5' to. So, kung saan mas malapit yung overhang, yun, 5', 5', 5', yan, 5', 5', rin itong mga protruding ends. And so question, which one of the following is a recognition site for restriction enzyme BAMH1? And so, balikan niyo. So, which one is the recognition site? So, for next, guys, is your restriction enzyme digestion protocol. So, this is just a way on just knowing that you could cut your DNA into fragments and you could visualize it using your gel electrophoresis. Okay, so once you have created your DNA fragments, you could now use it for molecular cloning. Sometimes the scientists will only digest the plasmid or vector using one enzyme. Pero minsan, naghahalo-halo sila ng enzyme to really cut it in smaller fragments. Okay? So, magbigay tayo ng mga example. So, this one is a plasmid DNA. So, notice that your plasmid DNA has two restriction sites for BAMH1 restriction enzymes. Yan. At, guys, pag expose mo tong plasmid with the restriction enzyme, it's called digested. Yan. So once this plasmid plus your BAMH1 restriction site enzyme, pinaghalo mo sila at niran mo sila sa gel electrophoresis, ito ang lalabas. Okay? So the number of restriction sites will be the number of fragments. So kung dalawang restriction site, di nahati ng gupit ng dalawa, di yung result, dalawang restriction enzyme na. So, kung tinanong ko, guys, how long, how many base pair is the total size of the plasmid DNA, simply add the two fragments. So, 4, 5 base pair. Yan. So, notice in the undigested, ibig sabihin it was not exposed to enzyme, 4, 5 base pairs rin siya. Okay? So, in this image, yung fragment nahati ng dalawa. So, 3, 2 base pairs and 1, 3 base pairs. Yan. So, you could expect na sa gel electrophoresis, yung isang band lalapat, yan, it will align on the 3-2 base pair. Yan, dapat hindi tapat. 3,200 base pair of the DNA ladder and 1-3 base pair of the DNA ladder. Yan. So, hindi masyadong precise, no? Pero, just a. So, magka-partner ang restriction enzyme digestion at electrophoresis. So, example na naman. Ito. Imbis na BAMH1, EcoR1 naman. So, same thing, dalawang restriction enzymes. So, expect na pag in-expose mo tong plasmid sa EcoR1 restriction enzyme, dapat dalawa ang fragment na lalabas. Okay? So, para malaman mo yung total number of fragments, simply add them. Yan. Or if not, since meron namang uncut or undigested, so, dapat 4,070 base pairs ang total nito. Okay? So, to check. So, how many restriction sites does EcoR have on the plasmid? 2. Okay? What is the total? 4,070 base pairs. Okay, next naman. So, this one rin, so this photo is also digested by EcoR1. So, let's look at ito ang A. I think this is the ladder, okay, or the standard. Itong B yung result. So the DNA fragments after EcoR1 digest. So my question, after seeing the gel electrophoresis, how many restriction site does EcoR1 have on the plasmid? If I told you the number of fragments is equal to the number of restriction sites. So 1, 2, 3, 4, 5. Yan. So 5 yung restriction sites, ibig sabihin, 5 yung fragments, ibig sabihin 5 yung restriction sites ng EcoR1 sa plasmid ng dinigest. Wala akong photo ng plasmid, okay? Gel electrophoresis lang pinakita. What is the total base pair in the plasmid that was digested? So, actually, itong first band, 2,000, so 2,000 plus 1,535. Ito, walang nakalagay. So let's assume this is 1,5. Ay, 1,5 na to eh. This is 750. 750 base pairs yan. Kung magpapa-exam naman ako, guys, bibigyan ko naman ng value. Pero looking at this, yan, 500, 250, mga 750 base pair to. So, 2,000 plus 1,500. Kuha tayo ng calculator. Yung ako na yung calculator ko. 3,500 plus 750, 3,250, 4,250. 2,000 plus 1,500 plus 750 plus 500. Wala na naman, 250 sabihin natin. Yan. So, kung ako gagawa ng estimate ng total base pair, it's about 5,000 base pair, okay? So, don't worry sa exam, bigyan ko ng value bawat DNA ladder. Pero, diba, kunyari, if you would just use your common sense, and then, you could get an answer about 5,000 base pair. Was the total plasmid that was digested by the EcoR1. Okay, next. So, walang gel electrophoresis. Ito naman, nakatabulate na. Sinabi na kung ano yung size ng base pair. Okay, sabi dito, 2 freshman college students perform the following set of restriction digest on a newly isolated plasmid BMLS 230. So, meron daw silang nakuhang brand new plasmid. Yan. So, they got a few enzymes and perform single and double digest reaction. Pag sinabi natin single, isang enzyme lang, restriction enzyme ang ginamit, like this one, HPA1, and HIN3, single digest lang. So, in-expose nila yung PBLA 230 sa HPA1, at ang result ay isang fragment lang, which is 26 kb or kilo base pair. Tapos, nung in-expose nila yung PBLA 230 sa HEN3, 1, 2, 3, 4. So, it produced 4 fragments. At nung pinagsama nila yung HPA1 and HEN3, double digest, 1, 2, 3, 4, 5. It produced 5 fragments. Okay? So, ang tanong, what is the total base pair in the plasmid that was digested? So guys, the total base pair in the plasmid yan, is the total of the base pairs per digest. So since the total dito lang sa HPA1, the total is 26 kilobase pair. Yan. So since isa lang yung gupit, isa lang yung restriction site, nung minove sa gel electrophoresis, ang result 26 kb. So, ito na yung size ng total base pair ng plasmid natin, guys. Ng buong size. So, pag in-add mo rin to, 13 plus 6 plus 4 plus 3, it will also result to 26 base pairs. Diba? Ito rin, 7 plus 6 plus 6 plus 4 plus 3, 26 kilobase pairs rin ang lalabas dyan. Okay? So, how many restriction sites does the REs have on the plasmid? How many fragments were produced? So, HPA1 only has one restriction site, okay? Because it only produced one fragment. I hope nakikits nyo na. In hint 3, so it produced 1, 2, 3, 4. So, it has 4 restriction enzymes. It produced 4 fragments. And HPA1 and hint 3, so nung pinagsama sila, it has produced 5 fragments. Paano kaya nangyari yun? So actually, mas malalaman nyo to gamit ang, if you would construct a restriction map, okay? Pero I just want to point out, kaya naging 5 nung pinagsama na, nung nag-double digest, kasi itong kung nasaan itong 13 kilobyte na to, nahati sa 2 unequal parts, naging 7, naging 6. Anyway, so, basta malaman nyo lang yan, yun na yun, okay na sa akin yun. So, kasi kailangan more practice to make restriction maps. Next, what is the total base pair in the plasmid that was digested? So, at least ito, lahat may tapat-tapat na value. Yan. So, sabi ko sa inyo, di ba, dapat the total, yung band, kung ano yung total nila, yun dapat, yun yung total base pair. So, dapat pare-pareho tong tatlong to. Okay? So, 5.3 plus 1.5, 6.8. So, 4.1 plus 2.7 dito, 6.8 rin. 3 plus 2.3 plus 1 plus 0.4. Dapat 6.8 pa rin ang labas na. Okay? Next, how many restriction sites? So, equal R1, in 3. So, nung pinagsama na sila, 4. Yan. So, hindi ba kayo na-weirdan? Bakit biglang naging 5? Yan. Di ba dapat 4 lang? Because merong site, yung kung nasaan tong HPA1, yan, nahati niya tong 13 kb. Ganun. So, madali lang to. Parang relo-relo lang to. Parang watch lang. Yan. Parang hahatiin ba lang. Parang pie. Parang pie siya. So, yung value ng buong plasmid is 12 kilobase pairs, kilobytes. So, what is the resulting fragment and fragment site of the following single digestion? BMH1. So, since yung mga BMH1, since isa-isa lang yung fragment ito, BMH1, isa lang naman yung restriction site niya. So, pag niran mo to sa gel electrophoresis, anong size ng resulting fragment? 12 kb pa rin. Okay? Kasi 1 lang yung restriction site niya. Wala nang iba. XHO1. Yan. So, notice dito, sa plasmid natin, isa lang yung restriction site. So, pag niran mo sa gel electrophoresis, ang resulting fragment is 12 kb pa rin. Isang fragment lang eh. So, kung nalilito ka, you could use this linear. Okay, and equal R1, since ang equal R1, yan, hinati niya sa dalawa, 2 equal parts. Yan. So, ibig sabihin, the resulting fragment, dalawa. Okay? And one fragment will have the size of 6 kb and one fragment will have the size of 6 kb. So, kung kunyari, hatiin natin sa 4 dito, 4 na parte itong plasmid natin. Diba? 3 yan. 3. So, pag inati mo further yung 3, divide by 2, 1.5 kilo base pair. So, ito na yung guide natin. So, madali lang kasi nga, alam na, ito yung mga given. Okay, what if nag-double digestion naman tayo? Pinaghalo natin yung EcoR1 plus BMH1. So, EcoR1, so pag EcoR1 at BMH1, ang resulting tatlong fragments. Kasi yung EcoR1, dalawang restriction enzyme niya, yung sites niya, yung BMH1, isa ang restriction site niya. So, dapat tatlo ang resulting fragments. So, first fragment is this one. Yan. So, i-add mo lang 3 plus 1.5 plus 1.5, 6. Yan. And the third fragment is 3. Kasi may nahati ng BMH1. And the third resulting fragment is 3 kb as well. Okay? So, ito, ang sagot dito. So, 6 kb, 3 kb, 2 kb, 6 kb, isang 3 kb, at isa na namang 3 kb. Okay? So, next, BMH1 and XHO1. So, BMH1 and XHO1. So, dalawa lang yung restriction sites ng tag-isa sila. So, dalawa yung fragment. So, ang resulting fragment, 6 kb and another 6 kb. Tama. Next, paano pag pinaghalo? BMH1 plus PS3 naman. So, pag kinat dito at kinat dito. Yan. So, 1.5 plus 3, 4.5 kb yung isang fragment. 3 plus 3 plus 1.5, 7.5 yung isang fragment. Okay? Okay. So, how about pag triple digestion na tatlong enzyme na? How about quadruple? So, pag lahat na nung quadruple na, ito na yung mga resulting enzyme. 1.5 ay fragment. 1.5, 3 kb, 3 kb, and 3 kb. So, bale, 5 fragments pag quadruple digestion. So, 4 na enzyme. 1, 2, 3, 4. Okay. How about triple digestion na? So, I hope nagagets ka. So, I gave more examples of restriction enzymes. So, pakiyo na lang be introduced, yan, acquainted. So, your BAM, kaya BAM, it's from Bacillus amyloliquefaciens. Grabe. So, COF1, Clostridium formicosceticum. So, bala na kayo dyan. And this one. So, ulit lang yung iba. So, familiarize nila yung sarili niya. So, yung tacrin, meron din siyang restriction enzyme. So, your DNA ligase, yes, your glue. Okay, so we have this molecular glue. So, this glue, itong DNA ligase, isn't it, that it's important in our replication for the joining of our Okazaki fragments, okay? So, what it does, yan, yung glue, how does it glue? It glue? Paano ba siya nag-glue? So, it adds a phosphodiester bond if there is a break in DNA. So, your DNA ligase, yan, may photo ba tayo dito? Yan. So, this one, it creates a phosphodiester bond if the cut is a blunt end, and it could create a phosphodiester bond if the cut is a sticky end. Okay, so it could only glue a complementary base pairing. Okay, so dapat kinat siya ng parehong, ito kinat ng EcoR1, dapat ito rin kinat ng EcoR1 para magkaroon. So once it's combined, yan, a foreign one and the vector one, yan, you have form a recombinant DNA. Okay. So, it could join both blunt ends and sticky ends. Okay, as long as the sticky end was cut by the same restriction enzyme. Sabi niya, up to 1 million breaks can occur in the DNA of a single human cell each day. So, busy ang mga DNA ligase. Yan. So, we also have a DNA ligase reagent. So now, andito naman tayo sa mga vectors. So vectors, example of a vectors are your plasmids, okay? These are the sites or the agents or are carriers, yan, that carry the DNA fragments, okay? So we could now edit these vectors. So also called as cloning vehicle. So ginagamit natin to as a means to cut and paste our genes. Yan. So there are two types of vectors. We have cloning vector. So ang specialty nito is to reproduce the DNA fragments. Yan, to clone, to be cloned, to be replicated, rumami. Okay? So plasmid, cosmid, bacteriophages, yung box, bacterial artificial chromosome, yeast artificial chromosome, and mammalian artificial chromosome. So hanggang box and yaks lang yung discuss ko, hindi na yung max. Kaya na magpasa ng max. So ang specialty nila, they could clone, they could multiply quickly. So they should have an origin of replication for them to have the ability to replicate. Dapat may mga restriction site, syempre, na pwedeng gupitin at i-edit. And we have selectable marker and reporter genes. So these are important so that you would know which one, if whether the plasmid was inserted to a bacteria. So kung nag-glow siya, kung may antibiotic resistance na siya, if you remember, if it could turn the colony to blue color, yan. So dahil possible yun dahil sa mga selectable marker and reporter genes. We'll talk about it later. At meron tayong mga expression vector. So ang mga expression vector natin, plasmid lang, okay? So they are good in producing now many proteins. So yun yung focus niya. It produces a large amount of stable mRNAs to be able to be translated into proteins. Yan. So magpo-focus tayo dito sa mga cloning vectors. Yan. So ang plasmid, ang advantage niya, it could be both cloning and expression vector. So what are the properties of a good vector? At lahat to ay nasa plasmid. So it should be autonomously replicating. So dapat it has the ability to replicate on its own. Hindi niya kasabay yung extra chromosomal DNA. It should contain at least one selectable marker. So these are markers which provide us, scientists, ability to detect which bacteria were transformed, di ba? So kunyari ginrow natin yung mga bacteria sa selective media which has ampicillin. Since may beta-lactamase gene yung plasmid, so yung sign para malaman natin na na-uptake yung plasmid, is it has resistance to ampicillin through your beta-lactamase gene. Same thing, another one that we learn in plasmid isolation. If the plasmid has beta-galactosidase gene, or also called as LACZ, so in the presence of X-gal in the plate, it should produce blue colonies. And the blue colonies will give us a sign that all those blue colonies had been transformed into the competent or transformed cell. So ang mga gusto natin, yung mga blue colonies, di ba? Hindi yung mga white colonies. So those are called selectable marker. Okay? This marker are important to detect if the cell are transformed. Yan. Or a transformant or a competent cell. So pag sinabi natin a cell is competent, it has absorbed. It has uptaken. Parang hindi okay yung absorb. Uptaken uptook the plasmid, pumasok sa bacteria yung foreign DNA. Next, it should have a unique restriction enzyme. So only one site for one restriction enzyme for different R is to insert foreign DNA. So it should have the ability to cut a piece yan so that you could insert your desired gene there where you got it. Yan. Dapat may restriction site siya. And it should be small for easy handling. So some of our vectors, ang disadvantage nila, they are large. Pero iba kasi yung gamit nila. They are large for a reason. So plasmid is small, okay? So advantage, it could replicate on its own. It has an origin of replication. It has a selector marker. Yan, selector. Explain ng selector. Selectable na lang. Selectable. Pwede natin. Meron siyang antibiotic resistant gene, for example, ampicillin or tetracycline, and one unique restriction enzyme na pwedeng gupitin. So, our plasmids, alam mo naman ang plasmid, di ba? So, example of plasmid is PBR322. So, this is the first artificial cloning plasmid. So, they made this in the laboratory, okay? So, if you're the scientist, you could use this as your vector, okay? So, kung may desired gene ka, pwede mong gupitin nito, open it, and insert your desired gene, okay? So, you could use your PBR322. So, P means plasmid, B means Bolvier, R means Rodriguez. So, they were the discoverers. So, may mga restriction sites, yung PBR32, itong mga blue, na pwedeng gupitin, at merong dalawang selective marker. So, meron siyang ampicillin resistance at tetracycline resistance. So, it has a BL gene, beta-lactamase gene, and tet-A gene. Hindi ko sure kung ano ng tet-A gene. Basta, it gives tetracycline resistance. So, kung yung bacteria, guys, it has absorbed or the plasmid has entered the bacteria, it should have both resistance to ampicillin and tetracycline. So, kung ampicillin lang resistance niya, no. Okay? Dapat dalawa. Ampicillin and tetracycline. And also, it has a repressor of primer, ROP. Yan. So, this one is the one that regulates the copy of the DNA. So, maganda dito sa ROP. Pero, pinalitan ko yung image eh. Yung unang image may ROP. So this one, dictates the number, the plasmid copy number, ilan yung copies. Hindi siya mag-o-over ng copy. Next guys, we have PUC19. So P means plasmid, U means university, C means California. PUC19. So it has LAC-ZA gene. Yeah. So this LAC-ZA gene produces beta-galactosidase. At pag yung beta-galactosidase sa plate, the plate has X-gal, yun yung tawag, or X-galactose sugar. So it will turn the X-galactose, it will turn the white colonies into blue colonies. So good na selective marker or selectable marker. And it will also give resistance to ampicillin. Tapos it has a polylinker, yan. So it is a multiple cloning site. So dito sa blue na to, maraming restriction enzymes. Ayan o. EcoR1, SST1, ayan, marami sa polylinker. So next, another plasmid. Ito naman is for plant cells. Mas maganda ipasok sa mga plant cells itong plasmid na to. So itong plasmid na to is from Agrobacterium tumefaciens. Itong bacterium to guys, once it encounters a plant, it could cause, its plasmid will enter the plant and it will cause a tumor in the plant, or ang tawag dyan is crown gall. Yeah. That's why TI plasmid means tumor-inducing. It creates a tumor, a swelling in your plants. It's from Agrobacterium tumefaciens. So this TI plasmid, so it's in a bacteria, which is Agrobacterium tumefaciens. It has a DNA or tDNA. So natatanggal to sa TI plasmid natin and it could enter the plant cell. So now this portion of the T plasmid, this tDNA or T strand, this is the one that enters your plant cell and cause the tumor. So ganyan na rin ang ginawa ng mga scientists. So they got a TI plasmid. They could edit the tDNA kung gusto nilang lagyan ng gene of interest. Yan. Yan. So they could, pinasok na naman sa Agrobacterium tumefaciens, and the bacterium is transformed with the recombinant TI plasmid. So now they could expose the bacteria to a plant. Tapos this tDNA, kung inedit nila to, could enter now the plant. And the plant could be transgenic or genetically modified. So, baka ito yung ginamit nila sa BT corn. Yan, pwede. Kasi sa plant, mas okay to sa plant eh. Yan. So, ito yung plasmid ng Agrobacterium tumefaciens. So, ang pangalan ng plasmid niya, TI plasmid. The scientist could edit the transfer DNA. And, the transfer DNA could be transferred to a new plant. Galing, no? So, it works for plant cells. If you want to genetically modify your plant. Galing, no? So, what else, guys? So, aside from plasmid, we have bacteriophage or your lambda-phage. Yan. So, your bacteriophage are viruses to, Okay, malilito. Yan. So the other name for the specific bacteriophage that could be a vector is your lambda-phage. Ito yung nasa module. So its advantage, it has more carrying capacity. Kaya niya mag-carry ng longer base pairs compared to your plasmid. However, it's difficult to clone. Mas madali paramihin ang plasmid excision. So, yeah. So, your bacteriophage could be a vector kasi hindi ba it could inject its DNA to a bacteria. Yeah. So, it could inject its DNA to a bacteria, di pumasok na yung genetic material na gusto mo. Okay? So, what else? Dumarami siya by hijacking the bacteria. So, magkalaban ang E. coli at saka bacteriophage talaga. So, guys, ma'am, paano i-edit yung DNA na nasa loob ng bacteriophage? So, sabi dito, 1/3 of the phage genome is non-essential, so it could be replaced by a foreign DNA of interest. So 1/3. So the 1/3, so this is the complete genetic material inside their bacteriophage. And galing, guys, they discovered na 1/3 of it is filler DNA or junk DNA, that it's non-essential for the bacteriophage. So what the scientists did is they could cut the filler or junk DNA with restriction enzyme, and they could replace it with a gene of interest. Galing niya. However, it could enter the complete DNA genetic material could only be packed or mapapasok sa loob ng bacteriophage if it has a size of 40 to 53,000 base pairs. Yan. So yun lang yung caveat. Dapat ma-achieve niya itong size so that the DNA could be packed inside the head or the capsid of the bacteriophage. So could we just have this terminology? So sabi namin natin conjugation. Yan, the plasmid is passed from a donor to a recipient through the sex pilus, correct? For when we say bacterial transformation, the bacteria takes up the foreign DNA. Yan, so ito yung ginawa natin sa calcium chloride, di ba? And through heat shocking, we transformed it. So once it's transformed, we call it a competent cell or a transformant. And guys, for the bacteriophages, the way that it could multiply or kunyari sabihin natin, infect other bacterial cell is through transduction. So DNA is moved from one bacterium to another by a virus. So if we use bacteriophages, it's called transduction. And mamaya meron pa tayong transfection. So other vectors are your cosmids and Bacs and Yaks. So your cosmids, best of both worlds, both plasmid and phage. So what's naging advantage nitong cosmids, or if the scientists would like a larger, could want to clone large fragments, cosmids, yan, ang gagamitin nila, okay? So it's not easy to handle because these are very large, okay? Pero depende sa use, okay? Advantage yun kung gusto mo ng large DNA fragments. So your Bacs, so it replicates inside the bacteria, it's circular. Your Yaks replicates inside a yeast cell. Yan. So yeast cell could be used as host and they are linear and talagang mas malaki pa to. So your Bacs and Yaks were used in Human Genome Project because it could clone as high as 100 to 3,000 kilo-base pair. So, hindi ako nagkakamali, up to 3 million base pair. And so, malaki talaga itong mga Bacs and Yaks. So, mahalaga siya ng Human Genome Project. Yan. So, how do we introduce the foreign DNA to a host cell? Yan. Or we call it transfection. So, introduction of vector into a suitable organism. So, yung plasmid, i-introduce sa bacteria. So, napag-aralan na natin yung una yung calcium chloride mediated and heat shocking, hindi ba? So, calcium chloride help us, di ba, in allowing the entry of the plasmid inside our bacterial cells. And we will talk also about lipid nanoparticles. Mahalaga to sa vaccines, especially on your Pfizer and Moderna. Yan, binalot yung mRNA gene ng lipids para makapasok sa katawan natin. We also have physical gene transfer. Pag-aaralan din ito lahat. Electroporation, gene gun. Yan. So that the gene could enter your plant, your cells, you could bombard them through a gene gun. Diba? Ang galing. So para maipasok. So guys, ang caveat dito, so, yun, maraming ways para introduce yung foreign DNA sa cell, pero talagang hindi lahat ng cell or bacterial cell could allow the entry of the DNA. Okay? So, meron rin tayong microinjection and ultrasound to increase the chances of the DNA entering the cell, and we have virus-mediated. So, parang AstraZeneca vaccine. They use adenovirus as a vector so that it could enter our body. Galing, no? So, kaya gusto ko i-discuss itong mga to kasi very napapanahon. So, let's start first sa mga physical gene transfer. So, first is electroporation. So, madali lang to electroporation. It uses low consistent electrical field. So the electrical field will produce nanopores, small pores in the membrane of the cell which would allow the entry of your recombinant DNA. So yung mga, kung di ako nagkakamali, itong mga green dito, these are your foreign DNA. Okay, sabihin natin super cold na plasmid. So, through an electric field, yan, the cell membrane becomes more permeable to the entry of your gene. Yan. So, dapat low, weak, at saka prolong yung electric pulse para makapasok yung foreign DNA sa cell. Yan. So, first is electroporation. Next is gene gun or particle bombardment or bioelastic particle delivery system. Ang galing, no? So, yung gene, nilagay nila sa gene gun. However, binalot rin nila ng gold nanoparticles. Okay? So, yan. Barilin. Babaril-barilin nila yung yung plant para makapasok DNA-coated gold particles. Yan. So that they will bombard the plant cells so that it would be transformed and would get the DNA. We have also microinjection. So yung mga ini-inject nila usually are oocytes, yung mga egg cells, fertilized cells, or early embryos to create your transgenic animals or your genetically modified animals. Actually, ginagamit na nila ito sa mga gene therapy. Yan. So, they would inject already a gene and in the nucleus, yan, to create to modify the gene inside the nucleus. So example, itong nanay na nanay na baboy, it produces higher higher volumes of milk. Yan, para mas maging mataba yung mga anak niya. So compared dito sa A and B, yung A is yung genetically modified one. So it produces more milk. So, itong B parang ano to eh, baka sa Pilipinas. Kaya to, yan. So, transgenic na daw or genetically modify yung this one. A cow. Cow A. Yan. We also have ultrasound. So, ultrasound could be used to increase the chances of the DNA entering the cell. Yan. So how does it allow the DNA to enter the cell? By creating cavitation, microbubble cavitation or cavitation. Yan. So this one, yan, yung ultrasound will produce a nanobubble, microbubble or cavitation. Tapos it will open up the pores of the cell membrane to allow the entry of the genes. We also have virus-mediated gene transfer. So what they use are viruses as vectors. Okay? As carriers. Yan. Carriers na. So dito, example, ito ay isang adenovirus. Tama ba ako? How do viral vector vaccine work? This is adenovirus and inside it is your is your spike protein gene. Yan. Tapos, papasok sa katawan. So, kung hindi ako nagkakamali, AstraZeneca at saka Janssen. Yan. Viral vectors ang ano niya, principle niya. Example, ito na naman. So, we have a P10 gene, PTEN gene. So, ito, pinasok yung PTEN gene sa adenovirus. So, PTEN gene, pinasok sa viral genome. It is an anti-tumor gene. Kung di ako nagkakamali. Yan. So, the adenovirus will enter the cell. Okay? And it will inject its genetic material inside the nucleus. Yan. So, dito sa AstraZeneca, Janssen, adenoviruses rin ang ginamit. So, ang adenoviruses, guys, siguro kaya ginamit yung adenovirus because it introduced the DNA into the nucleus of the cell, but does not integrate it into the chromosome. Okay? So, sa nucleus lang ini-inject. However, itong retrovirus, guys, what they do is they inject the genetic material directly on the chromosome. So, depending on the use. Pero kumbaga, parang mas safe yung adenovirus. Because vectors that integrate genetic material directly into the chromosome can cause errors and lead to cancer. Yan. So, mukhang safe naman yung AstraZeneca at saka Janssen. So guys, dito, yung Pfizer natin and Moderna, they are introduced in the body through lipid nanoparticles. Your mRNA is enclosed in lipid nanoparticles. Okay? So, bago lang to mga mRNA vaccine. So, nakagawa na tayo ng SARS-CoV-2 vaccines, yan, that are introduced in the body through lipid nanoparticles. So, gumagawa na sila ng influenza at saka HIV vaccines na covered with lipid nanoparticles. So, dito, ito yung lipid nanoparticle, andoon yung green sa loob, yung mRNA. That codes for your SARS-CoV, your spike protein. Ayan, galing, no? So next, guys, so we learned the different ways on how to introduce the DNA into the foreign cell. So yung pinaka-pinag-aralan natin is yung calcium chloride and heat shock. So cheapest yun and easiest to do. So selection of the transformed cell host. Yan. So this is the bacteria na hindi pa nakaka-receive ng recombinant DNA. So transformed bacterial cell with an altered vector. So, meron siyang plasmid yan, but hindi siya recombinant DNA. So, ang gusto nating bacterial cell yung merong mga recombinant DNA or competent cell or transformants. Okay? So, how do we do that? How do we select for the colonies? Napag-aralan na natin ito eh. So, by their antibiotic resistance and using the blue colonies, using a selective medium. Yan. So if the plasmid has selectable marker, it could help us, diba? Identifying which bacteria were transformed. Yan. So ito ay isang napaka-fast, rapid way on detecting your transformed cell and cheapest. Yan. So ito, diba? Pag nag-blue, kasi merong yung beta-galactosidase, na-convert niya yung X-gal. Ayan. We also have blotting techniques. Ayan. So, yung blotting techniques, siya yung reliable detection for DNA and RNA. But nung may PCR technique na, napalitan na siya. Napalitan na itong southern blot saka northern blot. But itong western blot, still it is used until now. This is the gold standard for HIV testing. Yan. So, for the detection of the proteins naman siya, itong mga blotting techniques. So, what are the steps on blotting techniques? First, you have to perform the electrophoresis para maghiwalay-hiwalay yung fragments. Then, you will expose the agarose gel to a membrane. Yan. Pwedeng nitrocellulose gel, nylon gel, ito, ayan, pwede rin nitrocellulose. Then you will expose it to a probe to help you detect it. Then you will visualize it to an autoradiogram. Mas lalo na pag ang ginamit mong probe is radio labeled, di ba? Yung autoradiography. So, yun, step 1, gel. Step 2, gene of interest is transferred and blotted or imprinted to a membrane, nitrocellulose or nylon. So, para dun mikit yung gene of interest, yung blot, yan, the membrane may be treated to covalently link the bonds para hindi matanggal. Then you will use a probe. Yan. Meron na ako dito ng invisible ng hybridization. So, hybridization means the process of base pairing between complementary stranded molecules. Kunyari, yung nasa blot, CCC, GGG, TTT. Yan. So, the probe should have GGG, CCC, AAA. Yan. So DNA naman. DNA. Yan. So, yan, and detection and visualization. So, if the scientists use radioactive labeled probes, then you could use it auto radiography. Sa protein, sa Western blot, HIV gold, di ba, HIV gold standard, they use immunostaining. So, itong dating technique, pinalitan na to ng RT-PCR. Okay. And how else do we know if the host cell was transformed through colony hybridization? So, we test the, so same rin eh, iba-blot, yan, tapos i-detect through labeled probes, tapos i-visualize, yan. So, luma na itong dalawang to, yung blotting techniques at colony hybridization. So, at saka yung calcium chloride, it's cheapest, most rapid, pero RT-PCR na lang rin. Okay, to see if our cell has origin of interest. And finally, guys, step 5, it's multiplication or expression of the introduced gene in the host. Example is for making your insulin. So, kailangan mo gumamit ng equipments to produce a large scale of the insulin. Yan. So, the bacteria, so you inserted the recombinant DNA, tapos, kunyari, meron kang blue colonies, kunin mo lahat ng blue colonies that will serve as your starter culture. Yan. So, we will place it to complicated machines for large-scale production para makagawa ka ng nakaboteng insulin recombinant drug. So, guys, that's it for today. Thank you for joining me. Bye-bye.